I've known him for at least 20 years now, I hate to admit it. We met early in our career scuba diving after a meeting that he held, an international meeting in Australia. He's a professor in Brisbane. He's been working on a number of eclectic, important scientific subjects over the years. He's a physical chemist, radiation biologist, radiation physicist, a biologist, and he comes to us with lots of these skills. In particular, now important to us is the studies that he's done of mitochondrial function and the mitochondrial genome. And a certain number of months ago, we had to write a paper on the role of oxygen in effects of radiation and the effects of radiation. And I didn't have a new topic. And I was talking to him, and he commented to me, you know, the mitochondria has 13 coded genes that are only in the mitochondria, they're not in the nucleus, they're in the mitochondria. And it occurs that, to me, that a lot of late radiation damage looks a whole lot like focal mitochondrial disease. And so we looked at this in some detail, we wrote a paper on it, and I asked him to do a second sabbatical here. His first sabbatical helped us get our U19 grant. And so welcome, Dr. Maguire. Thank you for coming back again. I look forward to hearing your talk.
Thank you. I'd like to start by thanking Paul and all the staff here who made my first sabbatical here so pleasurable, and have been making my second short sabbatical here very pleasurable as well. The weather here is a bit different to the weather in Australia. Let me first of all say, if you don't understand my accent, please put your hand up quickly, and I shall repeat the word, whatever it is I've said, if I can remember what I've said. Okay. As you know, there are many famous Australian actors and actresses in your country taking off prestigious prizes. There are a lot of Australian scientists over here as well. There's a lot more money in America to do science than there is in Australia. The honeypot is here for Pooh Bear. So I'm going to talk about mitochondrial DNA, and I've titled it Responses to Radiation, Aging and Xenobiotics. Probably not quite the right order to speak about it, in fact, and you'll see I've rearranged things slightly. And I've started off at a very low level of understanding, so I hope no one gets left behind in this ramble through mitochondrial DNA and mitochondria. Just to remind you, the organelle which I have a great love for is, of course, this mitochondria. This one, interestingly, Dr. Zhang, oh, sorry. This one, interestingly, is a human placental mitochondria. I did some work on placental transfer of various drugs and other things many years ago, and we just happened to put in some electron microscopy along the way just to show how healthy the placenta were. And it never struck me at that time that placenta actually had mitochondria. They do, and I'll talk a bit more about how many we have when we get on to some recent work that I've just done in the last week with Dr. Zhang. There are many ways to stain mitochondria, and I guess the other thing I want to talk briefly about is just how important mitochondria are to the publication, to the world of publications, sufficient that one whole volume of science was, one whole issue of science was dedicated just to mitochondria. And they, of course, as you can see, they're in every cell you can imagine. There's some there's some of my skin fibroblasts, in fact, taken many years ago when you're allowed to do experiments on yourself. I might just go back and just point out again that the shape which mitochondria take up within cells is somewhat different depending on the conditions of the cells and the type of cells as well. So mitochondria are generally reported as these oblate spheroid structures. You can, in fact, under real-time microscopy, look at mitochondria, and you can watch them associating with the cytoskeleton. They move along the cytoskeleton almost as if they're grazing off something being produced on the cytoskeleton, and they stretch out, they fuse together, and then they fall apart again. They are not there's no such thing as a mitochondria. There is a cellular mitochondrial population, and that's important for what I'm going to be talking about later on. There are wonderful ways to stain mitochondria. A friend of mine believes that what the mitochondria are actually feeding on is the products of glycolysis. They're going along. He has a theory that all the mitochondrial enzymes are arranged, glycolytic enzymes are arranged on the cytoskeleton in the order in which you've learnt your glycolysis pathways. If you have learnt your glycolysis pathways, of course. Some of you haven't, no doubt. So if I wanted to spend 3 or 4 hours talking about mitochondria, I'd talk about all this stuff. We know that mitochondria seem to be evolved somehow from an earlier form of bacteria which parasitized another cell and multiplied within that cell. And we can see from the human mitochondria that much of the material that came in with those mitochondria at the beginning of life, really, at the beginning of eukaryotic life, has been moved from the mitochondrial genome into the nuclear genome. Just to remind you of some of the important functions which still exist only in the mitochondria. The whole of the citric acid cycle, or the Krebs cycle, or the tricarboxylic acid cycle, depending how you learnt it, exists only in the mitochondria. One form of fatty acid oxidation exists in the mitochondria, another form exists in the peroxisomes. The electron transport chain, oxidophosphorylation, the major ATP source in the cell, exists in the mitochondria. Part of the urea cycle, 2 important enzymes of the urea cycle exist in the mitochondria. Mitochondria have a role in cellular calcium homeostasis, in plasma ammonia control, and importantly, more recently, we know they're a site of generation of reactive oxygen species and an important regulation point for apoptosis. Remind you that this is one form of extranuclear inheritance. One of the other great forms of extranuclear inheritance in our biological world, of course, is in chloroplasts. Chloroplasts have their own genome as well. The mitochondrial genome, I'm going to talk about homoplasmy, heteroplasmy and segregation, and I want to talk a little bit about some of the mitochondrial DNA disorders, both in terms of what we know as homoplasmic disorders and heteroplasmic disorders, and mention this threshold effect idea, a very important suggestion to explain some of the disease effects we've seen and therefore some of the effects we might see with irradiation. I'm going to talk about mitochondrial DNA and some of the mitochondrial assays briefly in terms of enzymes, sequencing, and then spend a considerable amount of time, I hope, on talking about deletion and depletion in mitochondria. So just for those of you who are engineers or mathematicians in the audience, we do know something about things like volume of mitochondria, about the inner membrane area, about the number of complex subunits, number of complexes of each type per mitochondria. We know something about how many electrons pass through per second. We know something about the amount of membrane taken up by the redox and ATP synthase complexes. And it's interesting that in rapidly respiring tissue, it's believed that they occupy about 40% of the inner membrane, a huge component. The mean distance apart has been calculated at somewhere between 70 to 200 angstroms. In other words, close enough to form microaggregates of complexes, electron transport chain complexes. The generation of reactive oxygen species, I said, is important both in terms of superoxide. These are generally toxic byproducts, or believed to be treated as toxic byproducts of respiration, and they accumulate if the early steps of electron transport chain are inhibited, particularly complex 1 and the coenzyme Q. For those of you that have forgotten where these are, I'll show you a pretty diagram in a minute about the electron transport chain. We know that sometimes there is a need to generate reactive oxygen species for destructive purposes, and in particular macrophages use this mechanism as a means of cell killing cells that they have that they are either about to consume or have consumed. But we also have inappropriate reactive oxygen species generation, and that's the basis of cell suicide. The regulation of apoptosis or programmed cell death. The primary step in the apoptotic cascade, of course, is the opening of the mitochondrial permeability transition pore, and that permits the release of cytochrome C, which activates the cytosolic caspase cascade. They also release apoptosis inducing factors, a flavor protein, which induces another pathway, and also some latent forms of caspases. The apoptosis is basically caused by excess calcium uptake by mitochondria, increased or overexposure to reactive oxygen species within mitochondria, or any decline in energetic capacity. And for those of you who are looking forward to a decent meal tonight, I'll remind you, whether you eat protein, carbohydrate or fat, all of the important energy components of those food sources lead generally via the citric acid cycle, which is within mitochondria, to produce NADH or FADH2, and those substances donate electrons and protons into the system, which, by feeding down the electron transport chain, produce ATP. There, as I promised you, is a pretty colourful picture of actually how that works. Electrons from NADH are passed down the electron transport chain. So we're looking at electrons, not the arrows, not the grey thin arrows, but the arrows here. There's a point for entry of electrons into complex 1. This happens on the matrix side of the membrane. Electrons pass through, losing energy, and finally pass from complex 1 onto cytochrome Q or ubiquinone, and thus onto complex 3. As these electrons pass through here, the energy deposited within complex 1 is used to pump one proton out from the matrix into the intermembrane space. And we can go on down the similar process happening at complex 3 and in complex 4, and finally the protons produced are able to come back into the matrix via this interesting complex 5 or ATP synthase, which is in fact nothing more than a tiny motor that rotates. So there's a rotor and a stator mechanism in there, and in doing so, in that rotation, is able to convert ADP into ATP. The ATP is translocated out by A and T, if you like, complex 6. The only complex which doesn't pump protons is in fact this complex 2. Complex 2 and complex 6 are in different colour from the others because they're the only complexes in this chain that do not possess any mitochondrial gene input. The subunits of electron transport chain in the ATP synthase then mostly are nuclear encoded. They must be transported into the mitochondria, therefore, so they all have a little tag at the front of the protein that says, I'm a mitochondrial protein, take me to mitochondria. They say take me to the inner membrane or the outer membrane or somewhere else as required. Some of them, however, the vital ones as far as I'm concerned, are the mitochondrially DNA encoded, and that explains extranuclear or non-Mendelian inheritance. Just to remind you, if you haven't heard it, all your mitochondria came from your maternal lineage, from your mother, from your grandmother, from your great-grandmother. And of course some people have used this as a means of tracing human evolution, and whether it is going to stand up in the long run or not depends on your point of view. Efficient functioning of any electron transport chain requires correct assembly of complexes in terms of position within the complex, correct orientation within the complex, and sites that are active. This is a picture straight from the web. This is a human mitomap, the mitochondrial genome map. There is a region here which is known as a low frequency deletion region, and a region here which is known as the high frequency deletion region. And the common 5 kb deletion region actually starts here and goes for about 5,000 bases round here, and is one of the interesting sights in terms of some of the research I've done in the past, and also some of the research we're doing right here at the moment. The 5kb deletion is one of a number of common deletions. It's called the common one because it was believed to be the most common one there. This one occurs at the site of some 13 base pair repeats at the beginning and wherever the end is. That 5kb deletion appears to increase as you get older. So there's the first link. Aging makes your mitochondrial deletion or your deleted mitochondria population higher. It also, however, seems to appear in some late onset dementias. In some work I did in Virginia with Davis Parker some years ago, associated late onset dementia such as sporadic amyotrophic lateral sclerosis, ALS, or in fact Alzheimer's disease, the sporadic form of Alzheimer's disease with this 5kb deletion. There is still discussion about whether that is absolutely believed by some people or not. I think the jury's still out. The 5kb deletion, how does it form? We used to think that there was a deletion here between these two 13 base pair repeats, and then that would link back up to here, and you'd have a smaller circle of human genome. But in fact, it turns out that the 5kb deletion is really a 5kb loss, 2 bits of 5kb lost from 2 mitochondrial genomes to give you effectively a duplication of the remaining 11 kilobases. So that leads to loss of, by whatever mechanism happens, it leads to the loss of a region of mitochondrial DNA from one or more mitochondrial genomes. And if you look back to that slide I had a few minutes ago, and I'll show you again down the line somewhere, a lot of very important biochemistry is going to be lost when you lose that piece of genetics. The way we actually assay this, and they still do the same today, we did this, as I said, back in 1996. We just simply designed primers on either side of the deletion, and we ran what is called a fast assay in PCR amplification. And by doing, if you leave this long enough, if you leave the extension time long enough, you'll get a huge 5,500 base pair piece of DNA being produced. If you do very short time PCR, and back then we had polymerase which wasn't very efficient anyway, and we could just give it 20 seconds extension step, and we would only get this small piece of DNA amplified. So we're looking for the presence of something which has a big chunk taken out of it. So some of the other features of the human mitochondrial or mitochondrial DNA generally, it is all circular. It contains a D loop which includes the origin of replication, and as Paul mentioned at the beginning, this piece of DNA codes for 37 mitochondrial genes, 2 ribosomal RNA genes, a 12S and a 16S, which are, if you know something about ribosomal RNAs, they're the types of ribosomal RNA which are characteristic of bacteria, not of the human nuclear ribosomal RNA. Human nuclear are more like 18 and 23S. The 22 transfer RNA genes, and those of you that know your fundamental biochemistry know that you only have 20 amino acids, so you really only need 20 transfer RNA species. There's only 2 extra, one's for serine and one's for leucine. And then there's these 13 subunits of mitochondrial complexes. Most of them, 7 of the 13, code for subunits within complex 1. 3 code for complexes within cytochrome C oxidase, and 2 for ATP synthase, and only 1 for ubiquinol cytochrome C reductase. The genome is very conservative of space in the human. First of all, it lacks introns. Many regions are read in both polarities. There's minimal transfer RNA genes, as I've already said. The stop codons are truncated, and they've exported effectively to the nucleus many genes which remain active in the mitochondrial genome of other species. So we have the smallest mitochondrial genome. And the other interesting point, of course, is that the mitochondrial genome of humans and most other species is not quite the same as the universal code. A further complicating factor is this issue of heteroplasmy. We don't just have one mitochondrial genome per cell. We have many mitochondria and perhaps one or two mitochondrial genomes per mitochondria. So if you have a mutant mitochondria appearing, it may start off with low representation within a cell, but it can, during cell division, end up with all of the mutated mitochondria, or it can clear itself of mutation. This allows for the phenomenon known as segregation or somatic drift between different lineages of cells. You can start off, as I said, with 50% or whatever mutant forms. You can end up either with completely cured homoplasmic or wild type. You can end up with a heteroplasmic mutant or a totally homoplasmic mutant form. Some people have linked this phenomenon to the onset of diseases that are caused by mutations in this genome. How do you assay for mitochondrial dysfunction? Well, I've said that there's this wonderful thing called electron transport chain. You can assay the individual enzymes, obviously, but it's time consuming, it's expensive both of reagents and staff. The results are sometimes equivocal. What source do you use for mitochondria? Do you use accessible tissue such as leukocytes, platelets, or do you use the tissues which seem to exhibit the greatest burden of mitochondrial disorder, such as brain or cardiac muscle, or less importantly perhaps skeletal muscle? There's always uncertainty about contamination with non-mitochondrial material such as cytoplasmic, other vacuolar material, or indeed nuclear material. Sequencing. We can sequence the mitochondrial genome. My friend Davis Parker in Virginia has spent about 5 years sequencing huge numbers of clones of mitochondrial genomes to try and find what the proportion of mutant load might be. The big problem there, of course, is heteroplasmy. It's time consuming, expensive, and everything else I said about the enzymes is similar, except that in addition you have the problem of possible contamination from foreign genes, either the technician or scientist doing the assay, the cell culture material, bacterial or viral contamination. Having said all that, people have been able to identify a number of different disorders linked to treatment, to therapies of various kind. Aminoglycoside deafness was one of the first such cases to be looked at. People going into hospital with a debilitating infection treated with aminoglycosides end up deaf, and it was always believed, of course, that the doctors or the nurses or someone overdosed those patients. It turns out that a good number of those patients who go deaf because they have a more bacterial type of ribosomal RNA present in their mitochondria. So their mitochondrial DNA genome has changed very slightly to look more like a bacterial ribosomal RNA, and that becomes more susceptible to those antibiotics. We all know about the involvement of things like doxorubicin in its various forms, inhibiting mitochondrial DNA topoisomerase, which inhibits mitochondrial DNA replication, therefore inhibits mitochondrial DNA repair, inhibits mitochondrial DNA expression. People who have been treated with daunorubicin can end up with things like hearts that don't function well anymore. AZT. I hope you recognise my. I've paid lip service to your language by saying Z rather than Z there. AZT, of course, is used as a potent treatment form for HIV and some other viruses. it inhibits DNA polymerase and therefore leads to depletion of mitochondrial DNA. I can list a whole heap more of these as well. At the other side, flip side, last year I had a PhD student who's now finished his PhD and gone on to a postdoc in Bethesda. We were asked whether we could try to determine whether some airmen who'd been climbing in and out of an aircraft which was fueled by jet fuel, whether or not we could test whether those airmen, sorry, Paul had to go to another meeting, I told him he could go, whether those airmen in fact had been somehow intoxicated and sufficient to lead to a mitochondrial DNA problem. The aeroplane I'm talking about is a thing called the F-111. Do you still fly F-111s in this country? You do. Okay, so they're nothing more than a flying fuel tank, and the fuel tank is made of segments that have to be sealed with various polymers, and it seems as though the exposure, when every now and again those seals have to be taken apart, cleaned up completely and resealed. It's a huge job. So they specifically employed very small people who could climb into the various tanks in the wings, up in the structures at the back of the airplane, and so on. It turned out that of the patients we looked at, and we looked at lymphocytes in these people, and we had to be very careful to get rid of any platelets because platelets are a rich source of mitochondrial DNA. We found that up to 80% of their mitochondrial DNA genome was depleted in that cohort. So therefore a very important occupational hazard associated with mitochondria. Before I start talking in depth about irradiation in mitochondria, I want to just quickly go over with you this idea of mitochondrial population statistics. I've mentioned in terms of heteroplasmy already. Just remind you that there are many copies of mitochondrial DNA within the cell. We normally only have 2 copies of each nuclear gene. And in some tissues, indeed such as a fertilized egg, you can have 100,000 mitochondria present. The idea of depletion is that you lose mitochondrial DNA from some of your mitochondria. Now I said before that mitochondrial DNA per mitochondria is a concept which is statistically tenable, but it's not factual, that once a depletion occurs in one mitochondria, that load will be spread throughout all the other mitochondria. The extreme case of mitochondrial depletion is a cell called the row 0 cell. We make row 0 cells by feeding them with ethidium bromide or some other compound which destroys mitochondrial DNA. So row 0 cells have no mitochondrial DNA in them, but they still have what's called mitochondrial ghosts, because most of the membrane materials are being produced from proteins encoded by the nucleus rather than by anything encoded in the mitochondria. So we still look as though you've got mitochondria, but they have no mitochondrial DNA. Therefore they can't make any of those electron transport chain subunits. Therefore they are crippled in terms of their electron transport chain and their ability to make ATP using ATP synthase. This has allowed me and a number of other people to put forward a technique for assaying mitochondrial DNA. In vivo, in vitro, or any tissue we can get hold of, we can basically do this. We choose a mitochondrial DNA target, we choose a nuclear target, and we just simply look at the ratio of mitochondrial DNA to nuclear target. Let's call that X. If you have a loss of mitochondrial DNA from any cell, then the value of that ratio obviously will be less than X. We can estimate X by saying, well, let's look at a cell with about 100 mitochondria with only one DNA per mitochondria, simplest case. Therefore 100 mitochondrial targets per cell. X is 100 over 2, there's about 50, and you'll see later that corresponds to 4 to 5 PCR cycles in a PCR assay. Here's how we go about assaying DNA at a very simple level in real-time PCR. So heat denaturation separates two strands of DNA, put a primer in at the 5' end. We add into the system some intercalator, which is a fluorescent substrate. As the primer anneals and the polymerase begins to extend the primer, then you'll begin to get binding of the fluorescent material in between the strands of DNA, and extension of this out to the end, of course, will produce a number of binding sites for that fluorophore. So in the cybergreen assay, the fluorescence doubles in each PCR cycle. And the target we're using at the moment is a region of about 526 base pairs, and was suggested by someone called Murphy, who works in Mother Cell's group in Dublin. I think I met Murphy when I was in Dublin 6 years ago now, almost 7 years ago, doing a sabbatical leave. I didn't meet any of the other players in this group. So they were trying to use the cybergreen assay to estimate mitochondrial deletion, and they simply chose this region because it was outside this often deleted region. It was in what they call the stable region of mitochondrial DNA, and they used that as one target, and the 18S ribosomal RNA gene in the nucleus as the second target. Murphy looked in fact at radiation effects. He's one of about 3 groups who've looked at radiation effects. I should mention that it's only in vitro that they've looked at this work. They've done no work on whole animal tissues yet. That's why I'm here. That's why we hope we can get some good results out of our lab while we're here, or after I've gone even. So Murphy and his group in 2005 irradiated some cells at levels from 5 milligray up to 5 gray. Now I know that looks pretty small compared to the level of, was it 16 gray we were considering this morning? Yep. Or 40 gray or 60 gray or something like that. But obviously it's still a significant dose. And even at levels of 5 milligray, they were able to, and at times but up as little as 4 hours and right up to 96 hours, they showed an increased deletion load. So more mitochondria with little bits missing out of them. But it turned out it wasn't the common 5 kb deletion. That's the first change from the iatrogenic and the age-related disorders I was talking about, and aging generally. Paradoxically, whereas we see depletion in most of the xenobiotic assays and in the tissues of people who have late onset disorders, neurological disorders, his results suggested an increased total mitochondrial DNA load. So not depletion of mitochondria, but actually accretion of mitochondria, increased number of mitochondrial genomes. The conclusions then, there is deletion there, so damage has occurred. Why are there increased mitochondrial DNA numbers there? Either because they haven't gone through that duplication process, they've just got single circles, smaller circles, which can replicate faster, or at an uncontrolled level, because they may have lost some significant control mechanisms. Or, as someone else has suggested, the damaged mitochondrial DNA are not being removed. Someone's forgotten to empty the trash. So we want to see whether this occurs in the types of tissues that are being used in the lab upstairs. So we're going to start off first of all by repeating Murphy's work on irradiated cells using deletion studies and depletion studies. We're doing that both on human cells. We're then going to extend that to mouse tissues. We're then going to develop a TACMAN approach, a quantitative TACMAN approach. The cybergreen assay which we've developed or have used here are basically semi-quantitative. They're qualitative rather than quantitative. The quantitation comes about because you have a TACMAN TACMAN 1 tube approach, in which you have 2 fluorophores being incorporated in a single tube. It cuts down a lot on the errors that you see in the cybergreen assay. We're going to apply that to both depletion and to deletion ultimately. We're going to apply it in vitro to mouse and human first of all. We're going to apply it in vivo to mouse, human, rat, dog, whatever other animal we can get, whatever tissue we can apply it to. And then we're going to apply it to tissue which has been rescued from a radiation damage. A fairly ambitious project. However, with the help of Dr. Zhang, we already have some results. And I've only been here 2 weeks. So this is results using the Murphy type design probes against human cells. This is just to show we can get amplification. Notice that the nuclear DNA spot is there in smaller amount than the mitochondrial DNA one. The other lanes in here, of course, this is a ladder of standards. This lane has got nothing in it. This lane has got another primer set just to show we could do PCR well. This is the placental DNA which we used. Note that we used total human placental DNA. Zhang then went on and did some real-time PCR using the cybergreen assay I described a little while ago. And this is the sort of results, not bad for a first attempt, I would have thought. These are our triplicate data when you'd refine it, just taking duplicates, much closer fit to data. And there's the actual values. The way you read these things, for those of you who are novices in real-time PCR, you define a threshold value as some level above the background level. And the threshold cycle is where your particular amplification begins to take off compared to other amplimers in the system. And you can see that 18S ribosomal RNA target begins to amplify later than does the amplification for mitochondrial DNA target. That means that mitochondrial DNA is present at a higher concentration than the nuclear target. So having done that, we read these figures off, and we're about 3 cycles apart, ergo a 3-fold excess of the mitochondrial target over the nuclear target. So all that says, if you think back to the figure I just derived before, somewhere between 4 and 5, we're not far off the mark, and more importantly it suggests in fact that placental mitochondria aren't there in great numbers compared to other tissues. The probe we want to design now is the TACMAN probe. I'll go through the TACMAN 1 probe assay first of all. As you said, as most of us know, 65 degrees, double-stranded DNA sticks together. The theoretical minimum number is 1 copy of mitochondrial DNA, of course, or any DNA. If you heat to 96 degrees, 95 degrees, you separate your 2 strands apart to form single-stranded DNA. If you put in some primers, then you will begin to you will bind to each of the strands. And if you design a TACMAN probe with a fluorophore here and a quencher at this end, that fluorophore probe will bind to one of the strands. It can be that strand or that strand, in fact. And then as you extend, the polymerase has an endonuclease activity, and while it's extending, it also is chopping up the TACMAN probe. So it releases then the fluorophore into the medium. The fluorophore in this state, bound to the probe, cannot fluoresce because of this presence of this quencher here, a process known as the FRET principle. So finally then you have a completely new set of DNA strands, twice as many as you started off with. You can take this one step further. The net effect, the concentration of free fluorophore also doubles in each cycle. An extension of that assay is a TACMAN-2 probe assay, in which you start off with one target, presumably mitochondrial DNA, another target, presumably nuclear DNA. Just apply the same thing using different primers at 5' and 3' end than you use in this target, obviously, and a different TACMAN probe as well. Two different coloured fluorophores. I've put them blue here or grey and purple there, but the same outcome. The outcome again is the concentration of each free fluorophore doubles in each cycle. The outcome is the same as before, that you can measure the amount of DNA in that system, and in this way we can get the ratio of one target compared to the other target. So what have we done then in terms of ordering stuff for our depletion assays? We have ordered everything for the Murphy variety of mitochondrial target and nuclear target. I've sat and designed mitochondrial targets for the forward and reverse for mouse, and the nuclear forward and reverse for mouse, using that cyber green technology, which is cheap and dirty, but it'll give us an answer whether it's worth going on. On the basis of what we've already done, I've gone ahead and I've also designed primers and probes for both the mitochondrial target and the nuclear target against the mouse. I don't need to design the human ones because we'll use the same ones that Nick Lintle used in that investigation of those airmen. And finally, we already have the Murphy type human deletion primers, and I've designed some mouse type deletion primers for there. The future. Well, what can we possibly think about down the line? I'm going to get really upbeat now because Paul has gone out, Professor Akuniev has gone out and bought for us a lovely new amplification thermal cycler. We can do lots of things now. We can look at 5 different colours. So this isn't anywhere near as much as we can do in the future, but it's a step in the right direction. If we have a primer, which we already have against us somewhere in this stable region, we have a nuclear target, we can get the representation then of that ratio to this ratio, will tell us something about depletion or accretion of mitochondrial genomes. If we design a target somewhere in a region which is heavily deleted or potentially heavily deleted, we can then do depletion and deletion in one tube on this new instrument. Okay, so I'm going to leave it there, I think. Oh no, I've got some conclusions, I should say. So, we know that mitochondrial DNA damage is a feature of ageing, and for people like Bruce and myself, I guess we have to worry about that. The rest of you can wait for a while. Mitochondrial DNA is a known target for xenobiotics, both the therapeutic ones and the occupational ones. Both of them appear to lead to depletion of mitochondrial DNA. Mitochondrial DNA is also a target for radiation in vitro. That's the work of Murphy. Is it the major target for irradiation also in vivo? We don't know. Are there therapies to protect mitochondrial DNA from damage? Irradiation damage to mitochondrial DNA seems somehow to be different if the work of Murphy and a few other people is to be believed. It appears to lead to increased mitochondrial DNA rather than depletion, up to 2-fold increase at 96 hours in fact. It leads to increased burden of deleted mitochondrial DNA segments. The deletion appears different from the common 5 kb deletion, a 4881 depletion, length of 4,881 base pair length rather than 4,997 base pair length of deletion. And this is across a 7 base pair repeat sequence that the deletion occurs. I'm going to leave it there and think about what I'm going to be doing in about 2 weeks' time and ask for any questions.